Hello, everyone. Welcome to SECOORA's webinar series, Coastal Observing in Your Community. Thank you so much for sharing your lunch hour with us. My name is Abby Wakely, SECOORA's Director of Communications, and I will be facilitating and moderating the webinar. SECOORA is hosting this webinar series to discuss, highlight, and raise awareness about coastal ocean observing activities in the Southeast U.S. SECOORA is the regional coastal ocean observing system for North Carolina, South Carolina, Georgia, and Florida. We are one of 11 that make up the NOAA-led U.S. Integrated Ocean Observing System. Our mission is to sustain and promote observations that help keep you safe. This is a quick snapshot of the technology we support to accomplish our mission. Here are a few upcoming SECOORA opportunities. We post all opportunities in SECOORA News at this link here. We have 2 funding opportunities available, and our annual meeting will be in Wilmington, North Carolina, June 18th through 19th. It is a chance to network with experts and provide input on climate and coastal hazard sections of our planning documents. Please be sure to register today. As a reminder, all participants will be in listen-only mode. You are encouraged to type questions for the presenter or technical questions for me in the bottom right-hand chat box. I will respond to them directly or post the questions to our presenter after his presentation. We are recording this webinar today and will be posting the recording along with the PDF of the presentation on the SECOORA website. We are so very excited to welcome Steve Morton from NOAA National Centers for Coastal Ocean Science. He will be presenting Monitoring Harmful Algal Blooms, the Power of Citizen Scientists, the NOAA Phytoplankton Monitoring Network. Steve Morton received his Bachelor's of Science and Master's of Science from the Florida Institute of Technology and Biological Oceanography, and his PhD in plant biology from Southern Illinois University. He is currently a research oceanographer with the NOAA Harmful Algal Bloom Monitoring and Reference Branch. During his career, he has authored over 120 peer-reviewed manuscripts, serves on the editorial board of 3 international journals, and serves as the technical expert for the United Nations International Atomic Energy Agency. Steve, thank you so much again for joining us. I'm going to go ahead and promote you to presenter.
Great, thank you.
Give me 1 second. All right, so it should be passing over now.
Okay, I just need to share my screens. Am I all set now? You can
I see your
my
screen.
title screen. Fantastic.
Perfect.
Well, thank you very much. I'm delighted to be here today talking about the Phytoplankton Monitoring Network. It's a program we've had within NOAA since 2001. It's using citizen science to actually monitor harmful algal blooms. I'm with the National Center for Coastal Ocean Science, which is part of the National Ocean Service, and with the Harmful Algal Bloom Monitoring and Reference Branch. So the first thing is, what are harmful algal blooms? A lot of people used to call these things red tides, but as you can see, some of them are not red, and they're usually not associated with tides. So the term harmful algal bloom actually was coined actually at the lab here in Charleston, South Carolina, probably 20 years ago. These blooms can actually be very toxic, and they can cause fish kills, mortality events. They can also cause human health syndromes. And as you can see, they can be anywhere from pink to green to even bioluminescent at night. So harmful algal blooms themselves can actually be lumped into 2 large groups. They could be either producing toxins or bioactive compounds, which can affect marine mammals and cause them to die and be sick. They can impact seabirds. And they also can cause human health syndromes. People can actually get sick, and there's some syndromes that can actually make people die. The vast majority of the marine harmful algal blooms that cause toxins are actually either dinoflagellates or diatoms. The diatom pseudonychia produces a compound called domoic acid that causes amnesic shellfish poisoning, and you want to put that in the back of your head because one of the 2 case studies I'm going to be talking about will be about the diatom Pseudonychia. The second case study we're going to be talking about today will be a dinoflagellate causes a toxin called saxitoxin that leads to paralytic shellfish poisoning. But there's a number of harmful algal blooms that are non-toxic, that they do not produce a bioactive compound, and they can cause harm to the environment or cause fish kills due to a number of different things. This slide is just to let me know, to tell you that all parts of the United States is affected by either a marine harmful algal bloom or a freshwater harmful algal bloom. And especially in the freshwater ecosystems, these blooms have become more intense and more frequently in the past 10 years or so. Especially the Great Lakes, Lake Erie has been having a number of harmful algal blooms that are cause huge problems and caused the city of Toledo to not have any fresh water for a number of days back in 2016. Within the National Centers for Coastal Ocean Science, we have two main branches that do harmful algal bloom research. These are our internal harmful algal bloom portfolio. We have a harmful algal forecast branch that's actually located in both Silver Springs, Maryland, and Beaufort, North Carolina. They actually develop forecasts for these blooms. There's two operational forecasts. One is actually for Florida, and one is for Lake Erie. That is that you can sign up for and actually get the forecast for these blooms. The forecast branch also develops new forecasts for different species. I am with the Harmful Algal Monitoring and Reference branch here in Charleston, South Carolina, that we develop different methods. The Harmful Algal Bloom Monitoring Reference branch, we have a number of principal investigators. One, Greg Doucette, has been working on different sensors to actually detect these harmful algal bloom species and their toxins using these large environmental sample processors, and also some handheld sensor development. We also have the analytical reference branch, part of our branch, that is the national response to harmful algal bloom events, and also to develop new analytical methods to actually determine these toxins. Another part of our branch actually looks to validate these methods and actually transfer these methods out into the field. And we'll talk about that a little bit later. We also have a part of our branch that also looks at mitigation and control of harmful algal blooms using different technologies such as ozone nanobubbles. But today we're going to be talking about the Phytoplankton Monitoring Network, the program that I'm the principal investigator of, that we monitor these blooms for an early warning of both marine and freshwater harmful algal blooms. So with the Phytoplankton Monitoring program itself, our main mission is to educate the public on harmful algal blooms, HABs, while expanding our knowledge of what phytoplankton exists in coastal and freshwater ecosystems. So, like I said, this program started in 2001 with 3 different groups here in Charleston Harbor. This was actually part of the South Carolina Harmful Algal Task Team that was actually sponsored by the Centers for Disease Control back in 2001. We actually grew this program with the help of 3 fantastic ladies. One helped me get this program into Georgia. Her name was Margaret Olson. And to expand us into South Carolina, a person by the name of Lundy Spence, and to North Carolina, Terry Kirby Hathaway. They were all part of a National Science Foundation funded program called Centers for Ocean Science Education Excellence. Without these groups in different states, we would not have been able to grow this program into a regional and a national program as fast as we did. We also got associated with a great organization called the National Marine Educators Association, NMEA, and they also have regional organizations within the state. Like in South Carolina, we have something called the South Carolina Marine Educators Association. They're fantastic groups to actually get people who actually use your research and can help you transition that into something that students can actually digest. So we've had a number of partners that helped us grow this program, included the Centers for Disease Control, South Carolina Sea Grant, and currently we have an interagency agreement with the EPA Office of Water that took this program into the freshwater ecosystems. So what's great about the program itself is that we loan you all the equipment. We give you we have the volunteers look at salinity with a refractometer. We give them a 20-micron plankton net that they do a 3-minute plankton tow. The thermometer to measure temperature. We give you gridded slides, so when you look at your sample underneath a microscope, you can't get lost within your sample. We give you all the bottles you need, and also a preservative. If you find something of interest, you can actually ship us the sample, and we could actually help you ID what it's there. So we have expanded our program. We're all coastal, and then we have a partnership and interagency agreement with EPA that we expanded the PMN project into freshwater ecosystems, where we look for 5 potential toxin-producing cyanobacteria. But for the most part, today's talk is all going to be on the marine side. So what happens when our volunteers are out there and they find a bloom? Well, they take a picture of it with their microscopes, either with a digital camera or their cell phones. They will ship us a 1-liter sample of the bloom water, and from there we can actually look at it with our advanced microscopy and our scanning electron microscope, which I'll talk about in a little bit later. From that 1-liter sample, we can also look at if there's a potential toxin that's actually there. So who are our groups? These are both some of our freshwater and marine sites. So our groups list everywhere from high school to middle school groups, college groups. We have a number of aquariums throughout the United States that are involved, different civic groups, homeowners associations, master naturalists. We have a number of state agencies involved. We also have other parts of the federal government, such as the National Park Service, who's also involved. This is slightly an older map. We have a lot more green dots that should supposed to be on this map. But you can see we're bi-coastal, and we're throughout the United States with more samples each year. So our training itself, since we're a national program, not all species are found in all parts of the United States, and we don't want a volunteer to actually ID everything that's there. That would be taking too much time. So we have regional targeted harmful algal bloom species. This is the species list of the different species we want to look at here in the southeast. There's a number of toxin and non-toxic bloom species that are actually found here. In the picture is my program coordinator, Jen. She is the person that does most of the training. It's usually done remotely, just like this through a program like WebEx or there's some other programs. We give you a background of what algae are, why some species are harmful. We give you the sampling protocols. We also give you how to ID these species. So this program works mainly because of different technologies such as WebEx. But also, if you're out in the field and you have a species that you do not know what it is, you can actually get on a digital microscope, and we could actually look at the sample with you via WebEx. We also have developed tools such as an iPhone app. And yes, we do have an app. It's called Phyto, P-H-Y-T-O. It's a free download app that you can get through both Android and Apple. Within the Apple has been upgraded recently, and you have a species list. And within the species list, you can actually get a number of pictures of that species. You can actually hit this bar here called pronunciation. You get the correct pronunciation of what the species is. We also have a dichotomous key based on shape can help you out identifying what species you actually have. And with the Apple app, you can also download your data directly to us. This link here to this YouTube video will give you more functionality of the app itself. But again, that is a free download called Phyto. The app itself was designed by one of our volunteers who actually happens to work for NASA. So we also have a great piece of technology in our lab. Most people can use light microscopes. The vast majority of light microscopes can only get you a maximum magnification of 1000x. Most people use it at 400x. But with something called a scanning electron microscope, we can get increased resolution. Like I just put up a species called Serratium and a scanning electron micrograph of the same species. And I'm going to increase the mag. So you have a species here called Dinophysis, a scanning electron microscope picture of that, a Cassinodiscus, and at a 2,200 micron, 2000x shot of the same species. And the last one is a diatom by the name of Pseudonychia that you have to use scanning electron microscope to actually ID what species it is. And this picture right here is at 20,000x. So with this scanning electron microscope, we can get better resolution than you can with a light microscope. What's also neat about this is that we have a number of school groups that can come through our lab, and we can actually put them on this scope too. So the Phytoplankton Monitoring Network itself, we train our citizen science to collect samples weekly or biweekly and to do a preliminary identification of what potential harmful algal species are actually there. With them, with their citizen scientists partnering with us, we can analyze these water samples for any potential HAB toxins. And then together we can identify temporal and geographical HAB trends throughout the United States. So what our data flow actually looks like is that you can actually, all of our data is downloadable. We have a web-based tool that that I will give you the web at my last slide actually is the website address. So the volunteers will enter their data via the web-based tool or through the smartphone interface. It goes to a database that's at Stennis Space Center that's administered through a part of NOAA called NCEI. You can download all of our data itself right from the database itself, or we have a search tool that you can do a GIS type map. And all of our data, of course, can be downloaded directly from our website or through this web portal called data.gov. So that's the nuts and bolts of the program itself. I want to give two case studies. One is a case study of different Pseudonychia species that's been found in the southeast. This is a diatom that produces the toxin domoic acid, which is shown right here. Domoic acid can lead to something called amnesic shellfish poisoning. And the second case study will be Alexandrium in Alaska. This dinoflagellate produces the toxin saxitoxin, which is shown here, which leads to a syndrome called paralytic shellfish poisoning. So this is a group called This is a group out of First Flight High School out of North Carolina. They have a group called the Phyto Finders. They were one of our first groups in North Carolina. One thing that we knew about amnesic shellfish poisoning is that it was common in the West Coast, but was not found in the East Coast of the United States until 2006. This group here found the very first Pseudonychia bloom in the East Coast of the United States in 2006. So we actually looked at our temporal trends from 2005 to 2010 from north of the Outer Banks all the way to northern Florida to see what type of distributions we actually have of this toxic diatom. The Outer Banks, so these graphs are going from north to south. As you can see, the vast majority of the red, which are the blooms, are actually found in the northern part of this study case. So what we've been finding is actually 4 different species of Pseudonychia: Pseudonychia pseudodelicatispa, Pseudonychia pungens, Pseudonychia multiseries, and Pseudonychia striata that's been found to bloom in the southeast. The vast majority of the toxins of the toxin domoic acid is actually from multiseries, seriata, and pungens. So not only did the First Flight High School find the first Pseudonychia bloom, this bloom was in 2006. This bloom was also found to be toxic. So it was found toxic both in the water and in blue mussels that were also collected at the same time. In fact, those slides I showed with the distribution, we actually did a paper in the Journal of Harmful Algae on the spatial and temporal distribution of this toxic diatom Pseudonychia in the southeast. But what was really neat is that the teacher at First Flight High School, Katie, is one of our co-authors on this paper. What was really interesting about this bloom is that there was a co-occurring mortality event of dwarf and pygmy sperm whales of the genera Kogia. So what we did is that we actually looked at some fresh samples. We also have an archive of different samples from 1997 to 2008 that we looked at, and 57% of these samples from the dwarf and pygmy sperm whales tested positive for the toxin domoic acid. We also had a paper on the domoic acid exposure in dwarf and pygmy sperm whales that was also found in the Journal of Harmful Algae. So what was really neat about this case study is that it was the first time that this diatom Pseudonychia was found in North Carolina waters. So this group, the phytofinders, are built up of a high school group, and this high school group actually won the North Carolina's Governor's Environmental Stewardship Award. Also, the instructor, Katie, she actually wrote a number of grants through IEEE. She gets, for the last 12 years, she's been getting up to $5,000 per year to fund her classrooms. And also, 4 to 8 of her students each year goes to Woods Hole, Massachusetts for a month to actually do some work. They also, IEEE also funds them to go to oceans. So like Oceans 18 was actually here in Charleston, South Carolina this year, and she had 4 students give 4 different presentations on their involvement with PMN. So the Phytoplankton Monitoring Network provides a window to research and response, which led us to the first identification of domoic acid in marine mammals in southeast waters. That was our first case study. Our second case study of how people use our information is Alexandrium or saxitoxin in Alaska. Again, this is a different toxin. It's a toxin called saxitoxin. It's caused by the dinoflagellate Alexandrium. So if anybody's been on a Alaskan cruise, this is where most of the cruise tracks actually go. It's southeast Alaska. So the Alexandrium in Alaska is a real problem, especially for tribal folks, because they use the resources as a food source. The state does not test shellfish except commercial harvested. So coastal Alaska natives have a 12 times more likely event to get PSP or paralytic shellfish poisoning than 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 the general public, mainly because they use it as a basic food source. So there's been a number of cases starting in 2010 of people getting sick. In fact, there's been deaths occurring in Haines, Alaska, which is in the upper part here, in 2010 of people getting sick on PSP and actually dying. In 2011, there was 21 cases of PSP. So the partnership we have with Alaska tribes arose from a common concern about this resource, mainly butter clams and hardshell clams and blue mussels. They've received no assistance from the Alaska state agencies because of funding problems. The coastline of Southeast Alaska is as large as the entire coast from California all the way to Washington. So there's a huge coastline and not much people that are there. So this program actually started with the Sitka tribe of Alaska, and they reached out to other Southeast tribes in 2013 to start this program to build tribal capacity to monitor for toxic blooms. So this is the top picture is actually our very first workshop we had. It was myself, a colleague of mine, Vera Trainer from the National Marine Fisheries Lab in Seattle. We had a person from Alaska Sea Grant. We had and some folks from the Sitka tribe itself. Our last workshop, we have a number of tribes from the southeast. We also have tribes from the island of Kodiak that are also involved. So this program itself has really snowballed itself too. So this is where the tribes are actually located at. It's all the tribes in Southeast Alaska are working on this project, and they're the main reason why we picked Sitka as the hub for the testing is that you can get flights from all these other tribes to Sitka within one day. If you've ever been to Southeast Alaska, you know that there's no way to get from, say, Ketchikan to Sitka without flying. There's absolutely no road system within the southeast part of the of Alaska. So this is the Southeast Alaska Tribal Ocean Research Program and where they're located at. So what does the monitoring that they do up there look like? Well, they do the phytoplankton monitoring methods, looking for the potential toxic species that are there. We have them look for mainly Alexandrium, Pseudonychia, and a species called Dinophysis that produces a toxin called oxalic acid. At the same time they do their phytoplankton monitoring, they also collect blue mussels. Those blue mussels are an indicator for a potential toxic event. They will also do filters of the water so they can actually see if there's any toxin that is in the water. They also, if you're a tribal member, will actually test your food source that you're actually collecting also. So besides the phytoplankton monitoring part we do there, we also assisted the Sitka tribe and to actually having a toxin analysis lab that's there. So they actually detect saxitoxin themselves using something called the receptor binding assay, which is a method that was developed here over the last 20 years. They measure domoic acid by a commercial ELISA kit, which is a lateral flow kit kind of the kin of a pregnancy test. So this allowed the tribes to establish their own shellfish management plan based on their sampling data. And lab is actually a central hub now for other research needs that the tribes in Southeast actually have, such as ocean acidification. What's also neat about this is that all their data is accessible through their website. So they have a really easy system of red, yellow, green, and gray. So red means it's above the regulatory limit of 80 micrograms per 100 grams of shellfish. That means do not eat shellfish at, in this case, in Sitka, at these locations. Green means it's been tested and it's way below the regulatory limit, and yellow means it's approaching. If you have a gray dot, that means that they have not had a sample there collected within a week, so you're on your own. So this is one way to disseminate the information. Another way is that they have public service announcements that they put on the beaches, where the mussel, where they're located for their licensing. They also put them in post offices, telling you that, like for this example here, from 4/9/19, this beach in Ketchikan had incredibly high numbers. So do not have any shellfish from this beach. They're considerably toxic. So this program has been going on for a number of years now. And as I said at the outset, in 2011, they actually had some deaths in Southeast due to people eating contaminated shellfish. Well, one of the outcomes of this program is that it enabled tribal communities to harvest this traditional shellfish resource safely, and to also mitigate any threats from the harmful algal blooms and their associated toxins. What was really neat is that during 2017, in the town of Ketchikan, there was actually lethal amounts of PSP that was actually found in the shellfish. Using the alerts from both the community-wide ones and also using things like the NOAA weather radio, alerts were actually put out, and there was no people getting sick or no deaths in 2017 due to PSP. In the first 18 months of operation of this program, more than 100 positive samples were identified. So this is a great case study of how we linked both the Phytoplankton Monitoring Network and toxin detection to actually help detect and mitigate the effects of harmful algal blooms. So how can you be involved? You can contact me directly. Here's my email address. You can also look through us through our website. You can also find us through Google, which is really an easy way. Just Google NOAA PMN, and we're the first thing that comes up. And we'd like to do special thanks to our funding agencies, NOAA, the Centers for Disease Control, the U.S. Department of Environmental, the U.S. EPA, Office of Water, the Bureau of Indian Affairs, and the ANA for all the funding that we actually have. And with that, I thank you all for taking your time out for your lunch.
Great. Thanks so much, Steve. What a wonderful presentation. If you want to go ahead and keep your PowerPoint up, we're going to open it up for questions. If you have any questions, please type them in the chat box, and I will ask Steve directly. We had a few come in during your talk, so I'm just going to go for the order that they come in. Marilyn asked, are there species of HAB that are predominant in California waters?
Yes, there is the diatom pseudonychia is real common. Also, you have a species of Alexandrium. So you have both PSP and ASP. In the freshwater ecosystems, there's a lot of different things. In fact, myself and Jen, my program coordinator, we're just asked to be part of the technical advisory committee for the California HAB program. So we're going to be doing some more work out in California. But yeah, so the coast of California has always been a hot spot, especially for domoic acid. That's where you have the sea lion seizing is in southern parts of California. Yeah. So yeah, there's a real problem. There's also the state actually runs a phytoplankton monitoring network there that's been going on since 1999 to give them a heads up on these toxin producing species also.
Great, thanks, Steve. We had another question. Are the volunteers doing size classes for pseudonychia, or is that only done at the NOAA lab?
For the toxin detection itself? Is that what the question was?
I believe so. This is from Becky. Becky, if you want to clarify.
Yeah, so we actually transferred the technology to First Flight High School to actually look at domoic acid using the ELISA kits. A lot of these toxins now, there are commercial ELISA kits that are out there that are commercially available, and the only thing you need is a plate reader and how to actually do it. But the problem with some of these toxins is that they have a number of congeners that the ELISA kits cannot pick up. So there's a real problem with multiple toxins in a toxin class.
Gotcha. Thanks, Steve. We have another question in reference to the case study in Alaska.
Yeah.
What are the regulatory limits for HABs? And then a follow-up, are these national limits or state defined?
These are all done with a partnership with the states and the Food and Drug Administration. There is something called the Interstate ISSC, Interstate Shellfish Sanitation Commission, ISSC, which is a partnership with all the states and FDA. NOAA's involved in that to actually set these limits. It's also where you get your methods to actually measure the toxins. So all the approved methods are actually been approved by ISSC and FDA for the marine side. So for PSP, the regulatory limit is 80 micrograms per 100 grams of shellfish, and that's set by ISSC and FDA.
Great. Thanks so much, Steve. We have another question from Jason. Has NOAA worked with or considered working with any drinking water utilities on HAB monitoring and planning?
That is actually under the jurisdiction of the Environmental Protection Agency, EPA, and they are doing exactly that. That was one of the reasons why the Office of Water wanted us to actually move our program, our citizen science program, from marine waters, marine and estuarine waters into freshwater ecosystems to give them a heads up on what species are there, what potential toxins are actually there. So yeah, so that's not under NOAA's jurisdiction. That's EPA's jurisdiction.
Okay, thanks for the clarification.
Sure.
And Robert, we will put the contact info on the screen again, and I'll go ahead and let everyone have access to Steve's email. 1 second.
Yeah. I went all the way to the start. There we go.
Great. Thanks so much, Steve.
Sure.
All right, we're getting some more questions coming in. How are marine and freshwater monitoring programs coordinated?
For our program, the Phytoplankton Monitoring Network, they're all coordinated through our lab here in Charleston.
Okay, perfect. Is your method for conducting cell counts using light microscopy available online?
Yes, there is. That's one thing that we do not have our volunteers do, is do direct cell counts. we have them do presence absence and or if there's a bloom. Nobody, I mean, cell counting is very difficult and tedious. So, but yeah, there's actually different flow cells you can actually purchase depending on the size of your plankton. One's called a Palmer Maloney cell, which is exactly 1 ml of water, and you count all the things that's in it. There's also a flow cell called a counting chamber called a Palmer Maloney, a Cedric rafter cell, which is a mil. There's also different technologies that are out there right now to actually count particles. One is called the FlowCam, which is an instrument that was designed by a colleague of mine up at Bigelow Laboratory for Ocean Science. It's now called in a company called Fluid Imaging. What it does is that it takes a flow of cells through a camera, and the camera takes pictures of everything that goes through it. So it gives you cell counts, it gives you this distribution. It's a really cool instrument. But there's also there's culture counters that you can get. So there's automatic things that you can do to actually do cell counts besides you sitting underneath a microscope with a counter and going 1, 2, 3, 4, 5.
Great, thanks, Steve. We have another question. This is your Lake Watery fan club. Are you still planning to bring Watery into the PMN this year?
We sure are.
All right.
Yeah.
And then we haven't. Go ahead.
So we are part of the South Carolina Harmful Algal Task Team, and Lake Watery is a very large reservoir in the upstate of South Carolina, and they have a consistent bloom of a cyanobacteria called Limbia. And Limbia was found last year to produce a toxin that's akin to saxitoxin called Limbia toxin. So we're going to be doing some more work up there. We're going to be doing work with the Homeowners Association with a group out of the University of South Carolina. Recently got an Oceans and Human Health grant. So we're going to be doing work up there. Stay tuned.
Awesome. Another question, is there a learning guide for STEM education programs for HAB monitoring?
There is actually a chapter. I co-edited a book called Harmful Algal Blooms, a consortium desk reference, and within that is a chapter on education that was written by myself and some folks up in Georgia, and in there is a lot of STEM type activities surrounded with HABs. Another really good place to actually look is through is a website called The Bridge. The Bridge is actually operated by, I believe, VIMS, Virginia Institute of Marine Science, and it's also associated with the National Marine Educators Association. But there's lesson plans and a lot of really neat tools that are actually there.
Great, thanks, Steve. Is there any information on the biochemical triggers to toxin production in the cell?
Boy, that's a great question there. There's considerable research being done right now to actually look at what type of triggers there actually are, both biochemically and environmentally, to actually trigger toxin production. So right now there is gene clusters we know of for such as the saxitoxin gene production and microcystin gene production in the cyanobacteria, but for the marine species, that knowledge is still lacking.
Great, thanks, Steve. For scientists studying HABs, what is the most effective method to promote these citizen scientist programs to get more citizens in these affected areas involved in the data collection?
One of the great ways of doing it is through the EPA regions. I know Jen just got back from a very large training course in Idaho. So we're in the process of going through different EPA regions to actually get more and more volunteers, and also through organizations such as the National Marine Educators Association, National Teachers Association. We've actually been getting the word out for that. There's also a I I actually belong to a consortium of it's it's through different federal agencies on citizen science. So we're getting the word out more like that too.
Great, thanks, Steve. Are state biologists encouraged to be involved as volunteers to aid their state and HAB programs?
Yeah, they sure are. We have a number of state agencies that are actually helping us out.
Great. And do you have any experience with measuring airborne toxins?
Airborne toxins. There is, I don't personally, but in our branch itself, we have done that before. There is one toxin in Florida called Bralvi toxin. That's from the Corenia bralvis, which is the Florida red tide, which produces an aerosol. So there's been a lot of research being done on how far this aerosol goes inland, what type of environmental effects they have, what type of human effects they actually have. A lot of that work has been done by Barb Kirkpatrick at Morton Marine Lab.
Great, thanks, Steve. And I think Barb has moved to GCOOS, the Gulf of Mexico Coastal Ocean Observing System.
Yeah, she is the executive director of that, but she's still at her office is still at Moat.
Okay. When you work with state departments of health or public health and link disease to source, is this data available through their websites or yours?
All the phytoplankton distribution data is available through my website, which is listed right here. So anybody can download a piece or all of our information. In fact, I know a lot of different agencies that have used our information not for the phytoplankton work, but for such as the salinity information. I know USGS has downloaded all my salinity information through the Southeast. And I know the Weather Service has actually downloaded all of our information in the Southeast to use that as a tool, as an indicator of climatic changes.
Great, thanks, Steve. And I'm going to try to state this. I don't know if I'll pronounce it right, but have you ever got toxin readings without having a bloom or even having a suspect organism? We have a lake that has toxin, but no suspect cyanos. I'll send this to you directly.
Most of the time you have the organism and then you can have the toxin. There are cases where the organism itself could be benthic. It could be an epiphyte that's not in the water column itself, so it could be on the sediment and releasing toxins. So with that case, I would have to look into it further. But for the most part, you have to have the organisms to have the toxins, of course.
Okay.
Or another case is that you could actually have you can measure the toxin, but the organism bloomed and died a week or so before you even got to it. So that's why it's nice to have frequent monitoring in the same locations.
Oh, that makes sense. Thank you. How is quality assurance of sampling process by citizen scientists documented for regulatory purposes?
So that's a great thing about us is that we're a non-regulatory lab. So, but seriously, one of the things that we have our volunteers do is that they collect a 35 mil sample, and that 35 mil sample is preserved and we archive it. So we can actually go back to any of our sample sites to actually look at exactly what our volunteers are seeing. Also, if there's a toxin-producing organism, they will send us a 1 liter sample, and from that 1 liter sample, we will do the cell counts and the toxin identification from that whole water sample. So any toxin that is actually found by our volunteers is actually the cell counts are done by myself and my group. The identifications are confirmed by my group, and the toxin itself is all done with our analytical response team. But
Great, thank you.
all the data itself is all QA'd, QC'd also.
Have you heard of using the HydroLab mini sonde, which can convert voltage into concentration of cyanobacteria in cells per milliliter? Is this a legitimate method for determining cell counts versus microscopic analysis?
Yeah, there's a lot of remote sensing type of devices such as that. The problem is that you can't tell what species it is. So you could have a bloom of species X that doesn't produce a toxin will look exactly the same as species Y that is toxic. So a microscopic examination is always needed to make sure that you actually have a toxin producing organism. Does that make sense?
Yeah, that makes sense.
Okay.
All right, and can you comment on cyanobacteria detection methodology equaling dipstick/EIA/LC-MS? I'm not quite sure what that stands for.
You know, I have to have more of a question there. I don't know exactly what those acronyms are.
Yeah, I do not either. Tdasku, if you want to email Steve directly, he can answer that question for you.
Yeah. If you email me, I can get you an answer.
Perfect. Thank you, Steve. And then we have one last question. What are the points of contact for NOAA for the Northeast coastline? Are they involved in HAB monitoring?
The Northeast in the New England states. We also have sample monitoring up there too. You can contact me, or if you're in the state of Maine has their own volunteer phytoplankton monitoring network, and you can contact the Maine Department of Natural Resources in Booth Bay Harbor, Maine, and the contact is, and I can't remember. They move on, so I don't. They have a marine biotoxins group there, so you can actually Google Maine Department of Natural Resources Biotoxins Group, and they will be able to get you to the Phytoplankton Monitoring Network up there. But if you're in the other New England states, we can help you out.
Great, thank you. We have time for one more question, and it is, how are advisories determined and shared when one of your monitoring groups detects a HAB that produces toxins?
Okay, so in the second case study, the tribes can do that themselves. However, if we find a harmful algal bloom, say in Charleston Harbor here, and we find a bloom of domoic acid producing species, I will test that, but we also will, at that same time, after we got the concentration, we will also contact the State Department of Health here. They're in South Carolina. They're the ones that would close the beaches. It depends on what state. So each state's different, and so we have a decision tree for each state. So if we have a bloom in Georgia, that contact we actually have, and if we have to contact them, we definitely will.
Great, thanks, Steve. Regina, we did not get to your question, but I'm going to link you up with Steve directly. And Steve, I'm going to take over. You're getting a lot of kudos. Great presentation. I thought it was really fascinating and really easy to follow. Thank you so much.
Sure.
And I'm just going to share one final screen so you all know where to get a recording of the presentation. A recording and a PDF of the slides will be available on this link here, SECOORA.org/webinar-series. For questions, please email either steve.morton@noaa.gov or communications@secoora.org. If we did not get to your questions, we'll pose them directly to Steve via email and copy you on that. And I do want to do one final plug. Help us kick off hurricane season starting June 1st with our next webinar, an overview of Hurricane Florence. It's going to be Tuesday, May 28th at 12 p.m. Eastern, and Stephen Pass from the National Weather Service Wilmington will be making that presentation.